this session, we are going to look for utopia for people with rare disease through innovations in clinical trials. And the first speaker that we have is Professor Gareth Balaam, who is the Medical Director of the Rare Care Centre, I beg your pardon, at Perth Children's Hospital, the head of the Western Australian Register of Developmental Anomalies. He is also a clinical genomics policy advisor at the Western Australian Health and founder of the Undiagnosed Disease Program, Western Australia. Professor Balaam has actually done a lot of work on rare disease and is the co-founder of the Undiagnosed Disease Network International and the Global Nursing Network for Rare Disease. And over the past 10 years or so, he has also partnered to implement AI solutions for diagnosis and care of rare disease across multiple health systems and multiple continents, i.e. the whole world over. And he conceived the artificial intelligence framework for rare disease. And the topic of Professor Balaam's is, I beg your pardon, unlocking treatment options, personalized in-time access. Professor Balaam, please.
Thank you for the gracious invitation to the organisers. It's really just been an amazing series of lectures. I feel so humbled to be here. And I know that my next speaker is far more expert in AI than I am. So you'll get the ridiculous followed by the sublime. And what I really... The title Utopia actually came from a discussion we were having with the European Commission as part of planning, this is about 7 years ago, as part of planning what would be the next wave that we thought of in rare disease. And really, we kind of sat there and we said, you know what would be amazing? It'd be amazing if, as soon as you had a diagnosis, it would link out to care and support, not just in healthcare, but in other sectors, disability, social services, education. Wouldn't it be amazing if that also pointed to novel treatments or linked you out to sort of clinical trials? And what would be really cool is if that did that in the absence of a diagnosis, because most people living with rare disease aren't diagnosed on our planet. And so most times we're dealing with combinations of phenotypes, signs and symptoms. So wouldn't it be amazing if we can design a system to do that? So someone in that sort of commission planning groups said, well, that sounds like utopia. Utopia. So then we retrofitted the acronym. So that's how we got there to the name. And we haven't achieved utopia. We're far from utopia, but I'm going to talk to you about a few steps along the journey. And very importantly, to acknowledge the real AI experts in here are not me, it's Professor Jutta Grozer, working together with Tristan and Shale and other really brilliant minds. And I want to also very much acknowledge Dr. Samir Jamur in Singapore for the collaborations we've done in Singapore with their electronic health record data in particular. This is a very stylised patient journey. Now, the patient journeys are never linear. They're very circuitous, especially for rare disease patients. But it starts with the inkling that this might be something wrong. The passage towards seeking or getting a diagnosis. Getting a diagnosis if you're one of those fortunate few that actually get the diagnosis. And then what's your care like? Your care in the medical system, your mental health, your physical health, your care in the education system, in the community systems, in disability services. And this is also a stylised version from a children's perspective because I started as a paediatrician. And so always that's my kind of default frame of reference. And it's also about transitions to adult care, transitions to employment, different sorts of transitions from the newborn out into the real world, into primary school, into high school, into middle school. And one of the challenges in the rare disease sector is there's so many rare diseases. And that's why they're collectively so common, because there's so many of them. But that also means that there's a challenge of low individual numbers per disease. So how do you actually get the knowledge to create these linkages, these pathways? And pathways that are personalised, that are predictive, that are preventative. And one of the great challenges, but also one of the things that absolutely permeates the rare disease world, and in a way is a strategic advantage, is that only 5% of rare diseases have a specific prognosis-altering drug therapy. That means for 95% of the time, there isn't a default to a drug treatment. So you have to think really holistically about your patients and how you manage them, because you don't have the safety net of a drug therapy. So an awful challenge for people living with rare disease, the lack of treatments, but also an amazing window into providing holistic healthcare. About 2.5 years ago in a meeting in Canada with the International Rare Disease Research Consortium and Genome Health and Genome Canada and a number of other stakeholders, we held a workshop on artificial intelligence for rare disease, really thinking about what would be a framework to deploy all these different AI approaches at. Some sort of international structure for this. And you'll be very unsurprised to see that there's a patient journey at the heart of that framework. And then thinking about the full range of services across primary through to secondary, tertiary and quaternary care, and the full range of sectors across government and our communities which deliver health and well-being, and I'm use the broader well-being lens here. And then what would some of the success pillars underlying some of these characteristics that are culturally responsive and that adapt to patients' needs? And that again informed what we were developing. About 12 years ago, we started in the diagnostic space and implemented a concept recognition and natural language processing in our public health system for rare disease across our health system. So we've been doing some stuff in diagnosis. Were we perfect? Absolutely not. Is there so much more to be learned? Absolutely. Are there people doing stuff out there that's better than us? Absolutely. But we started somewhere. And so as we've expanded, we still have some diagnostic focus, but we've focused much more now on care pathways and how we can use AI at every different point of the patient and family journey in a systematic way. So our current focuses are really much more on care pathways and access to care in supplement to diagnosis. Anyone heard of this? How good's ChatGPT? Well, better than me at advancing slides. So there's been several people look at ChatGPT and other large language models, both commercially available and bespoke models, and their suitability for the rare disease use space. The very high level summary of that is the common large language models that are commercially available out there have not been trained to work well in the rare disease space. I'm going to skip past this first one. No, I'm not. Sorry, I'm failing with this. So there have been a number of studies that look at using commercially available large language models, plus or minus tweaking them for rare disease. So I'm going to talk about some without the rare disease tweak. So this is a huge body of work done by Peter Robinson and many other famous people in the space, looking at the ability to use commercial large language models, in this case ChatGPT, and applying it to different languages. And the bottom line is that even with a relatively restricted set of data using ChatGPT, only 30% of the time did you rank the diagnosis in the top 10 possible diagnoses. If you train the model to think like you need to think in the rare disease space and to have access to rare disease data sets, it gets better. So this is work from our colleagues at PUMCH, so with Prof Zhang and her colleagues, where essentially they trained their models on case reports, rare disease case reports, and sort of taught the large language models to think a little bit like you have to do in rare disease. And by doing that, they markedly improved the capability of those large language models to rank the diagnosis in the top 1, the top 3, and the top 10. Essentially, they kind of doubled its ability. And then we kind of did it, learning from our colleagues that had done this work, doing some work in the space ourselves, thinking about some of these approaches. We thought, well, how can we kind of have test the large language models to say, how good are you at rare disease stuff? And so what we did was really say, okay, we've got these great knowledge bases, OMIM, Orphanet, Monarch, which are kind of sort of ground truths around rare disease that link up knowledge through ontologies and standardised terminologies. that create knowledge bases, let's test the large language models' abilities to build those. So let's say to those large language models, go and build us the knowledge base in Orphanet, OMIM, Monarch, etc. And we'll see how well you did. Not so great. So at the level of genotype, not too bad. They got about 60% of the way there. Still missed about 40% of the knowledge in those data sets. At the level of phenotypes, very bad. So at the level of phenotypes, they only recovered 10% of that knowledge. And this might be for a large number of reasons. Some of those reasons include just the lack of rare disease data out there that are influencing the base training of these models. And some of those things include things like genetic data is actually, in terms of genes and how they're influencing pathways and then leading on to disease, is much more accessible than actually the manifestations of diseases in people, their phenotypes, because that's locked up in case reports, often sitting behind firewalls, far less accessible information. So it's far less accessible to you and I, and it's far less accessible to the generalised large language models. So there's a number of reasons why we think this is the case. This just kind of says if you get a bit fuzzy about how you think, like humans do, and you say, okay, so something like an exact match, a non-fuzzy match would be macrocephaly, large head equals macrocephaly. A fuzzy match might be macrocephaly, microcephaly. That's a fuzzy match. There's a difference in the head size, too big and too small. It's not exact, but it kind of fuzzily matches. So if you kind of do this sort of stuff and build these into these semantic similarity searches, the models get better. We then also looked at individual diseases. So we did this across thousands of different rare diseases, and then looked at how the different models represented those diseases. And there was some overlap, but clearly also some difference. It really did depend on what knowledge base you were comparing it against, and also which large language model you used. But another take home from this is if you use multiple different large language models, you get a benefit. So multiple LLMs are better than an individual LLM. Okay, I'm just in the interest of time, I'm going to skip through some of these slides. Right, coming back to needles in haystacks, finding diagnoses, or what's the journey for that needle once you've found it? How does it get its guide? How does that needle get pointed by the magnet of its compass in the place to go forward? And again, when we're thinking about this, we also wanted some information from the diagnostic space and thought they're kind of new methods of kind of looking around this space. And so we looked at, we wanted to look into some population level data. So we worked with Singapore and used 1.2 million records from 1.2 million people to text mine for rare disease. Or in this case, what we did is we used SNOMED CT, which is a standardised coding system for clinical notes in electronic health records. So not the full free text of the notes, the coded data as coded by SNOMED CT, and then say, what can we find in these electronic health records, and how does that match what we expect to find? And so what we found are a number of different things. So, and then we wanted to have some sort of way of thinking about it. So we said, okay, we're going to group these different rare diseases, genetic diseases, cancers, non-genetic rare diseases. We might want to make them in those sort of clusters because that kind of roughly fits into the sort of like clinical pathways of who these people will be seeing. So let's kind of analyse it this way. And the largest group in that set were the non-genetic rare disease. Now, many of these autoimmune conditions, rheumatological conditions, etc., etc. And then rare cancers are obviously a very significant component. What we found was that about a third of the conditions match the prevalence you would expect from, in this case, Orphanet. And now we don't expect them all to match by any ways because there's lots of limitations of electronic health records, there's limitations on what that prevalence data, there's very little regional specific prevalence data for rare diseases, so we're very often matching against European data sets. So there's lots of reasons to suspect there'll be mismatches. But what we've mostly found is a huge rate of underdiagnosis. So a bit over half of cases were underdiagnosed based on prevalence alone. And that might have reasons, that might be due to differences to access to genetic testing. It might be due to differences in the disease profile in different countries, and also how diseases present, their phenotypes. Because the phenotypes of a condition might be different in different genetic backgrounds and in different communities and in different environmental circumstances. But all our medical education is biased towards what we know from essentially Caucasian data about how diseases manifest. So our index of suspicion for many of these diseases might be completely off. We then also wanted to think a little bit differently about trying to identify people in medical records. So there's some amazing work. I'm very impressed with the work done at PUMCH. I'm very impressed by the work with Think Rare and many other initiatives to sort of stratify for people potentially living with rare disease. So what we decided to take in this case was an approach which is now starting to be used a little bit more, which is an information content approach. And what that really means is how unusual is the data about an individual event? Or in this case, how unusual is the data about an individual person? So, and then a related concept, which is entropy, which is really about how diverse is the data. So you've got a marker of unusual and you've got a marker of diverse. And then, what we did is because we had serial electronic health records, we looked over multiple episodes of care over time. And what we found is that the rare disease patients they're unusual. And you can use markers of unusualness, I've just made up a word, to identify patients with rare disease. You don't necessarily need to know what their diagnosis is to find them this way. The other thing so that's that top line, basically saying these people living with rare disease are more unusual in terms of the information inside their electronic health records. And they get more unusual with more episodes. And it's not that, and why they get more unusual is they get more combinatorially unusual. It's not like you just add in more stuff and there's just more stuff there and it gets you over a threshold. It's actually, they have higher combinations of unusual things that accrue over time. The other thing about this was the reduction in entropy over time. So it always sticks out, in kids particularly, but also in adults, the diversity of the data is much greater. But what happens is they get less diverse over time. So it's almost like you converge down to common pathways. You start off really unusual with a very diverse spread of things and then converge down progressively into common pathways. And essentially in the adult sector, you get lost because it's harder to identify the rare disease in the adult sector because the diversity is less. So they're really hidden. In adults, people with rare disease are really hidden. This was just showing how these profiles change with time, again, sort of highlighting that it's easier, if you like, and to identify from an information perspective kids with rare disease than it is with adults. There's also a difference between genders, at least in this data set, and that with time, women become more have a higher information content, become more unusual and more diverse with time, and that in itself could be a flag. Then we then said, well, could you use this as some sort of screening test at a population level? And yes, you can use anything as a screening test at a population level, but how good is it? And so it turns out there's about a 95% sensitivity, so not too bad in a sensitivity, not perfect, but not too bad. And the precision, if you like, the positive predictive value is 20%. So you're going to get 4 false positives for every true positive with this case. So So essentially kind of double what Dr. Lau showed with the NPV, for instance, in that initial study, for instance. And it really was, you needed the multiple encounters to get to that precision. One encounter alone was not enough. Right. So nothing's static. We all evolve over time, our diseases evolve over time, our experiences, our environments evolve over time. And so we really wanted to create what we called computational natural histories or phenotype trajectories. How does a disease, or in the absence of a diagnosis of a disease, how does a set of phenotypes evolve over time? Can we create that as a natural history? Can we then use that to plan and prevent disease? And can we do that for all sectors, health, education, disability services, etc.? And so this is just showing a particular condition, it's an MPS disorder, and how that evolves over time. And the knowledge of how that evolves over time allows you to plan what interventions to do or not to do, and when to do them and not to do them. So if you know a child's going to go blind in X years, you can put interventions in the schooling environment, you can put interventions in the disability environment, as well as in the medical environment. So we thought, okay, how can we do this? Or at least how can we start this journey, because it's always an ongoing activity. So the first thing was to say, okay, let's go out to a bunch of published knowledge. Let's go out to some good knowledge bases, and let's ask the people. And let's use lots of different artificial intelligence processes, in this case, stacking up multiple artificial intelligence processes in a chain, combining multiple agents, and then having like a leading supervising agent to kind of help. So we combine existing knowledge that we could find from knowledge bases, from publications. This is Noonan syndrome used as an example, and then expert clinician knowledge. And we crowdsourced the expert clinician knowledge by exposing information from knowledge bases to clinicians, asking them to make linkages. What do you think connects to this? Which bit of this phenotype do you think connects to this particular intervention, or this particular outcome? And we use some of your expert knowledge to combine with the artificial intelligence processes. And then really was, kind of stacking up multiple AI approaches in a kind of chain to provide the information, to curate the information, to go back and check the information in different ways, and to use different large language models, because different ones are better at different aspects of the tasks. That then allowed us to build out these computational natural histories, these phenotype trajectories, how diseases evolve over time. So this is just a spider plot of how diseases manifest over different parts of the body, in this case Noonan syndrome, and how that changes over time based on the knowledge extracted from experts and from using artificial intelligence in those knowledge bases. Then we went back to medical records and says, does this kind of fit with real world data? So then we went back to records in KKH's electronic health record and said, how does this match with real world data? The heart of our Rare Care Centre is really about a holistic and coordinated approach to health and wellbeing. So our team is made up of people from all sectors, from the education system, from community systems, from the disability services sector, as well as health, mental health, Aboriginal health, etc., to really provide that holistic, coordinated view on healthcare. So we really wanted our models to reflect that, because it makes such a difference to our families. We've also shown that that model of care saves a lot of money for the health system. It's a 5-to-1 return on investment. Every dollar you spent on this care coordination model returns 5 back to the government. So really that's become our framework for building these models. And in Western Australia, we don't have electronic health records yet. Most of the world doesn't have electronic health records. We don't feel too bad. We feel bad. And so we've had to think about ways to deploy these that work with and without electronic health records, and that work with and without high compute versus low compute. How can we run systems on clunky hospital laptops? And so that's been part of our MO. And so we just thought about how can we deploy different parts of this chain within the existing health system, and how that can look in different health systems. So currently what we do is take patients' referral information, use multiple agents to then go out and do the research, which says build the knowledge base around this patient, build us a care pathway, and tell the clinicians the knowledge they need to know about this condition to have the best discussions with the patient. So they don't have to go and do all that administrative task of finding all that information. And they might not even know where to go. So let's do that for you and provide that to the clinician so that when they have their appointment, they can have a really high quality, precise appointment and use their resources really well. We then combine that from the information that then comes from the patient, and then present that information back in what we call visual summaries. So we thought very hard about how we actually present information so that the different service providers and the families can understand it and engage with it. That's kind of where we're at. And the things that we're really working on right now, we're very close to finishing off how we basically work in the disability sector with this information. So in WA and in Australia, there's a national disability insurance system, and you have to prove you have enough stuff going wrong to get access to these services, and you have to prove that the information you're given is of high enough quality for the people to make the decisions about whether or not they're going to provide the money to do the services. And you have to have some way of guiding the service providers about what sort of service they might want to provide. So what we've done is created ways to essentially auto-fill parts of those processes and provide validated information that says, this disease is associated with this functional consequence that leads to this need for this intervention in the disability services, and here's the level of evidence for that assertion. What we're working on at the moment is the clinical trials matching, because clinical trials matching is a big challenge. You know, for instance, when we looked at the first 100 patients that came through our care coordination service, only 2% of them were accessing a clinical trial in our state. Another 2% of them were accessing it either interstate or overseas, yet up to 47% of them were potentially clinical trial. There was potentially a clinical trial out for them. So there's a huge proportion of our families that are missing out on an enormous number of clinical trials just because the people and the systems don't know about them. Imagine if that was another domain. Anyway, agents. So how do we harness the power of all these multiple agents? How do we get them to work together with individuals like clinicians in teams? And really, ultimately, how do we do this in ways that lead to better pathways for care? Thank you.
Thank you, Professor Bainham. Thank you so much for your time. And now, see if we have any questions for Professor Bainham.
Thanks a lot for the great talk. I know for AI application, you really need a few ingredients. You have to ask the right questions, so you need researchers in the field. You need to have your AI scientists. And the third important thing on the work that you are doing is, of course, the data itself. So I'm very keen to have your word of advice and your experience in working with a patient record, in particular electronic health records. You have worked with Singapore, and you, of course, have done a lot of work in Australia. What are the important elements that we should look for when we are evaluating the electronic patient records in order to do good AI-related work? Thank you.
So garbage in, garbage out is one comment. And so I think still one of the potentially the single most important thing in improving rare disease care is getting rare disease specific coding in health data sets. Yeah? So be that orphanet coding, implementing orpha coding in your health data set. You get part of the way there with SNOMED. You don't get all of the way there with SNOMED. So we find that if the data sets are coded best in the first place, and ultimately you can do that right now by embedding orpha coding in your systems, that makes the most dramatic difference. It's then thinking about the curation of your data, what comes out, how you do that. And some accessible data is better than a lot of inaccessible data. So figure out what you can get your hands on and use that while you're working towards the Holy Grail.
Thank you. Another question from a lady there?
Hi, Professor. I'm from Tencent, and we are also pay more and more attention on the AI application in this area. So I have a question for you. As you previously mentioned, the performance of general-purpose large language models in handling data such as phenotypes is often limited. We've noted that some researches like phenotype GPT indicates that techniques like fan tooling or RAG can significantly improve their performance. But as these models already possesses a strong capacity for contextual understanding. However, the field of rare disease is characterized by imbalanced data and sold data, along with stringent data privacy requirements. So the question is, in your view, will this field eventually be dominated by general-purpose models, killing it all scenario like GPT-5, 6, 7, or will it maintain a degree of domain specificity with specialized models remaining in the hands of a few institutions like yours that possess the priority case data? Thank you.
That's lots of layers to that question, and thank you for it. What I would say is we would all like to be redundant right now. So we would absolutely love it if the general purpose models out there handled rare disease data really well. And I think that's the endpoint we want to be at. But I think there's a lot of steps towards getting there. And so I think for a good time yet, we're going to be working with rare disease specific models and how we make those better. I think one of the other critical spaces, and I think one of the key steps forward we've seen, is creating sets in rare disease for benchmarking these approaches to AI, things like rare bench, for instance. And so I think that benchmarking is going to be really, really important. My hope is that we get to the point that the general purpose models are trained up to handle rare disease. But let's think about this also from a more general cultural perspective. Our doctors are not trained to think about rare disease. In fact, they're actively trained not to think about rare disease. Think about the common solution. Think about the horse, not the zebra. And so a whole medical culture, and therefore the data that it's collected, how it's reported, and how it sits out there in the greater data systems and what the models are trained on, is completely perverted against rare disease. So I think part of this improving the knowledge base is actually is with medical schools. It's with faculties of health and broader sciences, and about how we train people about rare disease.
Thank you for all the questions, and thank you for your time, Professor Bainum. Now let's welcome Professor Lau back on the stage. And Professor Lau will be introducing our next speaker for us.
Our next speaker is Professor Yong Chen, who is a Professor of Biostatistics and Professor of Biomedical Informatics at UPenn. Professor Chen's research focuses on clinical evidence generation, machine learning and artificial intelligence, as well as the development of learning health systems. At Penn, Professor Chen founded the Pencil, Penn Computing Inference and Learning Laboratory. And he also established the Type 1 Center for Health, AI and Synthesis of Evidence, CHASE. And he reports directly to the CEO of the Penn Medicine Health System, an $11 billion enterprise, and he provides strategic advice on data-driven initiatives. Professor Chen serves as the statistical editor for the Annals of Internal Medicine, a statistical consultant for the New England Journal of Medicine, AI, and associate editor for both JASA as well as the Annals of Applied Statistics. He is also an elected fellow of the American Statistical Association and the American College of Medical Informatics. And Professor Chen's title of his talk this afternoon is Modernizing Rare Diseases Studies with AI. Professor Chan, please.
Thank you, Dr. Lau, for the very kind introduction, and thank you for having me here. So it's my great pleasure and honor to share with you what we have been working on this topic. So just very briefly about our lab. So we have been working on a number of areas, very much centered around the so-called electronic health record data, but more broadly, so-called real-world data. So our lab has been very fortunate to be funded by quite a number of large consortia. So currently we are funded by about $60 million US to lead a number of national and international studies. And this is our lab member, so we consider us as not just in the field of statistics or AI, we want to consider us as a think tank to tackle important public health and biomedical problems. And there are three areas we have been taking deep dive. The first one is the so-called PANDA project, which is on the left, this one. And the goal is trying to leverage national database from different health systems to build early detection algorithms for rare diseases. So this is involving as many as 35 million patients over the time. And the second project, which is a $12 million grant about the data coordinate center, we are actually supporting 13 different studies. Each study was given about $20 million to generate multimodal data for us to develop computable phenotype for so-called deep phenotype for different mental health conditions. The last one has a lot to do with Brian's question about the data quality, because we all know garbage in, garbage out. So in particular for Alzheimer's disease, the goal is trying to build an ecosystem and the so-called common data elements engine to normalize this multimodal data for clinical research. So our lab, as I mentioned, started with a more statistical focused lab, but now we are very much focused on clinical studies. So this is basically, this relative proportion is basically showing how we devote our energy. We spend about 50% of the time truly diving into different clinical questions and taking the lead to tackle those questions. And on the other hand, those efforts actually help us to generate meaningful, novel settings to advance statistical machine learning AI methods. So back to some introduction about the real-world data. FDA has a formal, has been publishing guidelines for the use of real-world data. And a number of keywords is reliability and generalizability, and those ethical considerations. Those are very important considerations. And more recently, FDA also published this document about regulation of use of AI in these biomedical studies. And these topics include synthetic data generation and how to handle some type of different types of biases and heterogeneous data and so on. So we recently worked with FDA people to, and we developed 2 papers to talk about the practical consideration of using those medical records, insurance data for drug development. And one of the most useful figure is this figure it really covers the whole life cycle of use of real-world data from patient recruitment. Now, the corresponding AI innovations called the patient matching using large language model, single-arm trial, and then that motivated people to study digital twins, and data integration, and label extension, and drug repurposing. So this diagram is very specifically tailored for real-world data consideration for rare disease. So today I'm going to actually touch a number of those topics and show what we have been made, effort we have been made. Okay. So this is the PANDA study. The PANDA is an acronym came from my collaborator, Peter Merkel, who is chief of the division of rheumatology. And so the goal is to use this national databases to collectively develop this early diagnosis algorithms. And a practical challenge is the data privacy. I think in the morning we also talk about the privacy, especially this is a concern for rare disease patients. So we have 2 major databases. One is from Florida, which contains about 15 million patients. It's a centralized database. The other is so-called STAR network with about 20 million patients, but those data are decentralized across different hospitals. And we have to use something called federated learning to combine the evidence together to build those predictive models. And before we develop a federated learning algorithm, there is a we actually considered a number of potential rare diseases, the GPA, and those with abbreviations over there on the top right corner, and the eGPA. Some of them are conditions in rheumatology, and some of them are actually not as rare as we thought. For example, PSA. Psoriatic arthritis has a prevalence about 2% in the general population. But those are very useful use cases for us to develop a phenotyping algorithm and build predictive models. So the lesson learned in this effort are the following. So first, we need to admit the fact that there's no single algorithm being perfect. Regardless of how hard you train your phenotyping algorithm, they come with specific sensitivity and specificity, right? And the second fact is that typically you can have multiple algorithm-derived phenotype with different sensitivity and specificity. And so we really need to have clinicians involved to conduct charter review to understand how those algorithms perform. And ideally, we want to combine this machine intelligence with this human intelligence. Then the question is, how do you do the chart review? Obviously, for rare disease, you don't want to do a random chart review. That would be a waste of money, right? At Penn Medicine, we pay clinicians about $50 per chart. So if you ask them to random sample 200 patients to review, it's a waste of money, right? So this actually involves some technique called related to active learning. You need to design enrich the cohort with more cases, then that's better use of your budget. The second thing is about how to optimally combine the machine-generated phenotype with your chart review. That actually leads to a new framework, which we call the surrogate-powered semi-supervised learning. So a group of students has made a great deal of effort. My first student tackled working on this problem, Jesse Tong, and just joined Hopkins. That's where I graduated from last year. And there are 2 other students working on these problems. So those are some considerations in terms of data quality. And another important consideration is about dealing with all kinds of biases in the data and also integrating data from different resources. So a good example is this so-called recovery initiative. So in the United States, the second year of the pandemic, the government allocated a great deal of resource, about $1.2 billion, to sponsor 3 consortia. And we were leading one of those 3 consortia with a focus on studying pediatric long COVID. And there are a number of questions on the bottom right, as you can see. So we want to characterize those long COVID and understand the real-world impact of vaccine on the COVID infection and the long COVID. And those are actually much rare compared to adult population. So using real-world data, actually, we have developed a number of studies to answer this question. So this is an Annals of Internal Medicine paper we published trying to using causal inference to quantify the Pfizer's vaccine's protection, real-world protection against the COVID infection, and also how well this protection reduced over time. And the second one is the people want to understand what is the protection of the vaccine against the long COVID. And and also characterizations of this COVID and the long COVID conditions. We have a sequence of paper targeting different body systems, organ systems, from neurological to GI and so on. And the most recent one published at Lancet Infection Disease was a study trying to, that we found that the reinfection among children, the reinfection of COVID actually is causally associated with doubling or tripling the risk of developing long COVID. So last month I was interviewed by New York Times and Time Magazine just to elaborate this type of message. As you know, in US, there's a political environment against having children to take a vaccine. So this is what I think is a really important message to make a societal impact. So, so if I actually followed this, the chat channel about all this, how people view our findings at the New York Times. The first one, as you can see, you see people truly taking this seriously about the impact of vaccine. We need to vaccine the kids. On the other hand, they have a lot of skepticism against the real world data, right? The second person registered an account called Common Sense and said, for some reason, I just don't believe this, right? So, so this is actually tied to the next thing I want to talk about, which is how can we improve the reliability of real-world data-based studies. So just like the general topic in the morning, we talk about we need to collaborate, right? We need to share data across different countries, different data partners. So this is actually a very good example. A consortium called Odyssey, Observational Health Data Science and Informatics. This is a consortium established about 10 years ago now, connecting about 500 million patients across the world. As you can see, this international study, most likely you cannot share your patient-level data to a centralized place. And what we need is that we need to develop some type of protocol distributed to different data partners and ask them to do some analysis and then send the result to us. So that's literally the definition of federated learning. But the real-world federated learning, we need to care about some concept called communication efficiency. Because when we run a number of studies like this, imagine if you recruited 20 data partners and your federated learning algorithm need 3 iterations to get your final result. And quickly you will run into a situation called synchronization. Because some people may drop the ball and delay their task. And then you will be facing a decision. Are you going to drop this data partner, or are you going to wait for everyone to get their result before you proceed? Right? So that actually highlights the better option. If we are smart enough, we can develop an algorithm that only needs one communication, what we call the one-shot communication to get the final result. So just have a thought experiment. So in this room, suppose you are leading a study with 20 data partners. Would you be, there are 2 options. Option 1, each data partner only need to run this algorithm for 20 minutes, but you need to have 3 iterations. You need to have everyone finish their task, update your model, and then before you proceed to the second step. That's option 1. Option 2, you can let the computer to do all the work, let the computer to run this algorithm for 10 hours, but you just need 1 round of communication. So how many of you would prefer option 1? I don't think many of you would prefer option 1, right? Because we can let the computer to do the job. So that is exactly the reason for us to develop this suite of algorithm, which I think would be very useful for our rare disease studies called PDA. So far PDA has supported more than 30 clinical studies, and there's a lot of pediatric study and adult study covered using this PDA algorithm. And one really good example is this collaboration with IARC. Some of you may know it's a part of the WHO. This is called the International Agency for Research on Cancer. So they were running our PDA algorithm, and my collaborator Vivian on the top right from Paris, he contacted me, asked us to customize a solution for their study. So they have this study called the EPIC study. Basically, the EPIC study is it's just like a UK Biobank, but it's a very cancer-focused study where they want to identify the biomarkers that are predictive to the progression of different types of cancer. Some cancer are really rare, some are relatively common. So a practical issue they run into, which is also very common in Asia, is that some countries are willing to share patient-level data, even some blood sample data. Some countries, like Sweden and Norway, they cannot share their patient-level data, right? And another practical challenge is that for cancer study, people typically don't use a traditional cohort design. They use something called a case cohort design, meaning that because you don't want to collect the blood sample to all the controls. That's a waste of your money. You want to be, cost effective. So you collect the blood sample for all the cases, and then you also collect the blood sample for a selective group of controls to be cost effective. And it turns out that they cannot use our earlier algorithm, like we developed 5 years ago. This was from my first student working on this topic, Ray Duan, who joined Harvard Biostatistics as a faculty after graduation. So we had two algorithms called ODEL and ODEC. One is for one shot, which means only one round of communication, distributed algorithm for logistic regression. The other is for fitting time to event data. But those algorithms were devised for cohort study, not for this cancer case cohort study. And turns out that with my current postdoc, we were able to quickly modify this algorithm, but I really want to show you how it worked in practice. So here we have data, actually pooled data from 4 different countries, Italy, Netherlands, Spain, and the UK. So basically we can pool the data together. We can run some model. For each of the protein, we can get some so-called hazard ratio. So here we scan 7,500 proteins. So we actually have 7,500 estimates. And now we can pretend the data are decentralized. Okay, pretend they are decentralized. And then we can run our federated analysis. The first analysis is actually just meta-analysis, right? Everyone, most of us know how to run federated meta-analysis, just divide and conquer. And turns out that the pooled analysis result is fairly consistent, as you can see here. They are generally quite consistent with meta-analysis results. But if you think about the identification of biomarker, the sensitivity is 84%, precision is 92%. Not too bad, right? But now if you use a federated learning algorithm divided by us, you can see the mean square error is much smaller. And if you think about the classification of biomarker, the sensitivity is 99%, precision is close to 100%. So basically, we were taking advantage of the fact those 4 countries have the data shared. So we already internally tested this algorithm. And then we are currently deploying this algorithm to include Sweden and Norway. So as a side topic, we are currently working on an APAC project, working with data from China, and Australia and Japan. Some country has a more strict data sharing policy like China, and some country can share patient level data. It's exactly the same type of problem with the solution that we customized for WHO. So beyond this algorithm, we also developed a number of tutorials to educate the data partners. For the sake of time, I'm not going to play this video. So this is a web page. I think it's an industry level. We spent at least $500,000 building this user-centered data sharing open source framework that people can use this to run our algorithm. It's self-guided. It's called PDA OTA. OTA stands for over the air. It's something I borrowed from the Tesla. They have this OTA function, right? Over the time you can update. So when I was preparing for this talk, Brian asked me to talk a little bit about drug development, how to use real-world data. So So I think one way to consider this is actually very simple. So it's, in my term, it's basically X, Y, Z. So if you think about in real world data, X is a drug, Y is an endpoint of interest, Z is your population, and actually everything, almost everything you saw in the literature can be put in this framework. So label extension, which is basically means that you fix the treatment and outcome, you're trying to identify broader Z population. And the drug repurposing, you fix the treatment and the population, and you want to find additional outcome of interest. A good example is the semaglutide and the tirzepatide, right? People are using that to repurpose for other conditions. Initially treating diabetes and weight management, but now people are using that to treat even heart failure and kidney conditions. And also people find some signal for Alzheimer's disease. I think this type of consideration could potentially guide some related considerations in rare disease, because FDA has a usually so-called fast-track approval for rare disease. And then this could be some incentive for the pharmaceutical company to consider relabeling their indication. And the last one is the drug identification. For example, if for Alzheimer's disease, which is known to be very difficult, we can fix the endpoint, which is Alzheimer's disease, and then population is usually older than 65, then you can scan a large number of treatments trying to understand which drug could be potentially repurposed for the Alzheimer's disease. So one of the examples in particular for this drug indication extension is extending the indication of GLP-1 from adults to adolescents. I can show you one video about how we adopt this collection of the tools in AI to automatically help a trialist to design a new trial. So this is a 2-minute video I'm planning to play. So,
welcome to Trial Map. Randomized controlled trials are the gold standard for evaluating new treatments, but specifying inclusion and exclusion criteria to determine eligible patients is often complex. After regulatory approval, treatments enter real-world use. These real-world data capture much broader patient populations. For example, in weight management, GLP-1 receptor agonists were initially approved for adults, and they may also be safely extended to adolescents and young adults or pregnant women. In oncology, therapies approved for one tumor type or biomarker may also show promise when extended to other tumor types or molecular profiles. In these post-market settings, substantial real-world data can inform future trial design. TrialMap is an AI-powered data-driven co-pilot that guides users through 4 simple steps. Choose a cancer type, pick a trial, define the endpoint, and review eligibility criteria. Some criteria, such as ECOG performance status, cannot be relaxed, while others can be set as fixed or kept flexible. TrialMap then emulates alternative designs using real-world data. It generates multiple pathways, each defined by a unique combination of eligibility criteria. For each pathway, TrialMap evaluates complementary metrics, treatment efficacy by hazard ratio, safety by adverse events, feasibility by the number of eligible patients in EHR data, and additional measures assessing validity, generalizability, and efficiency. Origami plots, an alternative to radar plots, visually illustrate the trade-offs among these metrics. Pathways can be ranked using an uncertainty-aware composite score or an individual metric of interest. Start your journey now and make clinical trial design broader, faster, and smarter.
So this is one of the examples we can directly leverage EHR data to help with the trial design. So for the sake of time, I'm going to just skip some detail. But in the second framework of this drug repurposing, we have developed a number of tools like real-world evidence transformer is basically a debiased causal transformer using this adversary training idea to improve the reliability of the result. There's another concept called unlearning, recently getting more popular in AI, in the sense that there are lots of so-called spurious correlation learned from the neural network that you actually can use some human knowledge to help you to prune those additional unnecessary connection in the neural network. That has been shown to be very, very effective. So both papers are currently still under review at Nature Machine Intelligence or Nature Medicine. And there's another thing, as people are talking about, agent. There's we all know the agent or the LLM can have this hallucination, right? One of the strategies is actually design a system to have them to leverage each other. Then the question is, how do you define a good mechanism to help them to leverage each other? It's better than majority vote, right? You already saw some failure in United States about voting by itself, right? So what are the mechanisms, right? This is actually the place you can introduce something like called game theory, a Nash equilibrium to learn. So there's a lot of interesting part at the intersection of statistics and AI to develop this causal AI. So I'm going to just directly jump to the concluding remark. So this is the second to the last page. So with this federated learning algorithm, what we can do now is actually have this so-called online real-time evidence generation by running this study across different data partners. And I think at the end of the day, this is really the key figure I want to show. When people talk about AI, often people are thinking AI as some type of a black box, right? It's flexible, but it's acting like a black box. On the other hand, there's a concept in getting popular in pharmacoepi called target trial emulation. Basically, using real-world data to emulate a trial, you can speculate. So it's transparent, but it's not as flexible. And then on the left-hand side, there's a concept called a negative control outcome. That's some type of human knowledge. For example, from our human knowledge, we know COVID vaccine is not going to protect you from a flu infection, right? That's already known. So those type of a pair with no causal association is very good, very powerful prior knowledge to help you improve your reliability of the study. So all of the AI tools we develop, we are trying to hit the intersection of all three framework, which is interpretability, reliability, and flexibility. So I hope those tools can be in the later days that, when we have the discussion, we can discuss in more depth about how to leverage those tools to help with rare disease study and drug development. So thank you.
Thank you very much, Gareth and Yong, for two wonderful talks. And I didn't actually raise my hand to ask a question to Yong earlier. Just perhaps I can start off by asking you a question. You sort of ended your talk by mentioning the real-world data center and how useful it is. In fact, I know that in Hong Kong, we are also going to establish a real-world data center as well. I mean, what are the key elements of success, to a real-world data center? I mean, what advice do you have for us?
Great question. So I think the number one thing is about the infrastructure, right? So as Gareth said, especially for rare disease, you need to make sure you capture those patients. And there's a concept that we call the fragmentation, meaning that a health system only capture a segment of the patient journey, right? So we need to understand how patients migrate over time. So those are the important considerations, like data fragmentation and how complete the data elements are. Can we build a reproducible infrastructure for different researchers to access the data while maintaining the data privacy? So those are the first wave of questions coming to my mind. And the second thing is about engaging those downstream users early. You don't want to build something and then ask other researchers, say, is this what you want, right? We want to engage potential user at time zero. So those are the things I learned over the time. So it's infrastructure is everything, but we really need to bring all the stakeholders together to plan.
Okay. You actually touched on a very important point, and that is actually the engagement of the downstream users. Obviously, the two of you are very successful, experts in what you do, and I'm sure in many departments, institutions, or whatever, the clinicians don't always talk to the bioinformaticians and vice versa. When you have obviously been very successful. Could you share with us actually how you started, when you first started? Perhaps I can start with Gareth and then Yong afterwards. When you first started, what were the obstacles? Were there any obstructions actually from your partners? And what were the carrots and sticks that you actually have to encourage people to actively participate in your work, and then, eventually with the fruition that you have got now? Gareth?
I think the North Star concept is really important. So what I mean by that is responding what the patients and families, the people living with rare disease and their families are asking us to do.
Yeah.
And what their needs are, because nobody can actually argue with that. It's the one thing that no one could say, well, you've got a vested interest. We yes, they do have a vested interest in their lives and the lives of their families, and how can we deny that? But I think if you're going as a clinician, as an academic, as a health system executive, and you have a particular point of view, you can always open to some sort of criticism. That's just life. And so I think the more that patients and families, people and families are at the front of everything we do and are guiding what we do, and that there is a single voice from that community. I think that's particularly important with high-level decision makers, that there's a single voice from the community talking. So there's a harmonised question, because then as soon as the discussions are 5% different with a key decision maker, that's enough for inaction. And so I think that's critical. I think realising that rare diseases are things that every clinician is already dealing with all the time, and that the solutions require all of us, and respecting that existing expertise and how we can work together, and recognising that we simply can't do it without that cooperation. And then it's probably just years of building trust, and not over-promising.
Trust is very important. Yong, you have...
Yes. So before I answer, can I ask the audience, how many of you graduated without a degree in medicine, say in quantitative science, computer science? Could you raise your hand?
How many of you are not medical?
Not from
quantitative
medicine.
science.
Yeah, we have at least 15%, right? 20%. So I graduated from Hopkins School of Public Health, and then there's a in our profession for biostatistics, there's a saying the biostatistics in School of Public Health versus biostatistics in medicine. Those are two different cultures. Because some of you may know, in School of Medicine, there's a saying, School of Medicine is composed of MDs and everybody else, right? So, so I think my point is that for statistician or any data scientist, the critical thing is to find a research data partner with good mutual respect. So one of my main data partner, he said the most dangerous person at Penn Medicine is MD with a SAS license, right? So you really need to have people with domain knowledge to help you to work together. We need to understand, it's not one person's work. It's really at the intersection. We need to work together. So I think that's the most important thing for for us to work together across discipline. We need to respect each other and try to build something bigger than ourselves.
Thank you. Well, I dare say that the person that you just mentioned, of course, is great, but in many faculties or universities, we don't have that many of these people. I think training the next generation of, for me, clinicians who understand AI is very important. But then we only have so much time, actually, to train our students. What advice do you have? What should I do, actually, with my medical curriculum? How much AI should I teach my students? And what aspects of AI should I teach my students? Gareth, yeah?
As usual, I'm going to modify the question. I would say, how much rare disease are you going to teach? No, seriously. And so I think what we're seeing amongst us now, what we're seeing is that rare disease is a discrete medical domain that needs a dedicated focus, just like you have discrete medical domains for cancer or for cardiovascular disease or insert domain here. And so I think we need dedicated programs that are focused to rare disease and the unmet needs of people with rare disease, and are actively teaching the processes of how to approach rare disease. Because as I said before, right now medical schools are actively teaching us out of ways to approach complex problems, and in particular rare disease. So I think there needs to be a fundamental change in medical education that is focused to rare disease as a domain rather than focused to a particular technology. And I think then things will flow out of. Then I think things will flow out of that, and we can't. And I think also thinking about that sort of interdisciplinary intersection. A lot of our sort of advances or our partnerships are with the mining school, or they're with the business school. Yeah. And so I think those interdisciplinary partnerships are really critical, because often the problems that we're seeking to solve have been solved in other domains. We just don't know about them. And so I think that's that sort of interdisciplinary collaboration, I think will be key. So I think you need people with broad sets of understandings about the key principles that underlie rare disease, and then individual people will have that kind of T of depth, and then we need to connect across the sectors. I think that's part of the solution.
Thank you, Gareth. Yong?
Yeah, so when I graduated from Hopkins back in 2010, my advisor, Kun Yi Liang, he also left Hopkins for a president position in Taiwan and called Yangming University. And we had a conversation. I asked him, "What's your final advice to me?" And he said, "Never fall in love with any particular model." So
he was talking about your professional life. Yeah. Just checking.
So now I'm giving similar advice to my student, because AI has a faster iteration. When people talk about, 5 years ago, people don't talk about AI, they talk about big data. Then people talk about data mining, and then machine learning AI. Now it's agentic AI, multi-agentic AI, right? So I told my student, don't chase the wave, okay? I think, the wave come and go, but you need to understand what are the underlying principles. The principles actually is only a handful of principles, say reproducibility, understand the biases in the data, and you need to understand what is uncertainty. Those are key principles, but with different magnification, right? So I think those are the mindset I'm training my student. I think those are the critical thinking, really position our student very, very competitive in the job market, as you can see. So I'm kind of just reinventing this version of the final advice from my advisor to my advisees. So,
thank you both very much. I know that our time is up, so my last comment to wrap up this session is, first of all, we must thank our two speakers for the wonderful talks and also for the discussion. And I would like to very quickly thank the Hong Kong Genome Institute for organizing this symposium. Fantastic, and we have a very good turnout.